The human body is characterized by an extraordinary ability to correct internal biological errors and to repair injuries due to trauma, disease, or aging. In some circumstances and in some organs and tissues, this ability fails and the damage becomes permanent and irreversible. The objective of the Mat2Rep project was the development of innovative solutions to enhance or restore the self-healing capacity of mammalian organisms in two specific clinical conditions: traumatic spinal cord injury and diabetic foot. In the first case, the lesion results in motor and sensory disabilities, involving different body segments up to tetraplegia. In the second case, a simple skin wound opens the door to a progressive process that can result in limb amputation. Mat2Rep has developed new therapeutic solutions for these objectives, tested for safety and efficacy in preclinical models. This was made possible through the participation of doctors, chemists, pharmaceutical chemists, biotechnologists, veterinarians, thanks to the numerous technologies used and to the contribution of companies of the pharmaceutical, electromedical, materials, and biotechnology fields. The first therapeutic goal of Mat2Rep is the treatment of the traumatic injury of the spinal cord. There are no therapies for the acute phase of the injury, and the functional outcome, in other words, the residual locomotor and sensory sensorial capacity, is not to date pharmacologically modifiable. Starting from an experimental idea developed by researchers from the Interdepartmental Center for Industrial Research, Life Sciences and Technologies for Health at the University of Bologna and at the IRET Foundation, Mat2Rep has developed a multi-drug therapy, including controlled drug release systems based on nanoparticles, aimed to mitigate the damage during the early stage of the injury. Nanotech Lab at the Department of Life Sciences at the University of Modena and Reggio Emilia has designed, produced, and characterized new nanomedicines built from biodegradable and biocompatible polymers to deliver an anti-inflammatory drug, ibuprofen, and a promyelinating agent, thyroid hormone T3, in the spinal cord. Nanotech Lab has also developed a new conjugate of two anti-inflammatory drugs to target multiple pro-inflammatory pathways and optimizing stability, dimensional homogeneity, and the surface charge of the new nanomedicines. The formulation aspects were then taken care of and fully optimized to protect loaded drugs from instability, guarantee their pharmaceutical properties, and a prolonged release kinetics for 14 days, therefore appropriate for the clinical problem. These new nanomedicines were then tested at the University of Bologna and IRET Foundation, both on cell systems and in the spinal cord lesion model in laboratory animals. The new nanomedicines penetrate target cells, favoring the differentiation of the oligodendrocyte precursors, the cells that repair the myelin sheath covering the axonal extensions of neurons, to ensure the correct transmission of electrical impulses. T3-loaded nanoparticles were then included in the multi-drug therapy in spinal cord injured rats at the IRET Foundation laboratories. By using a battery of sensory motor tests conducted with automated and non-invasive methods, we showed that Mat2Rep multitherapy improves the functional outcome, and in particular the integration between sensory and motor signals for a proper locomotion. A synergy with the molecular and cellular biology, proteomics, and image analysis laboratories also allowed to verify the biological effect at a tissue level. These results confirmed the experimental hypothesis. Mat2Rep multitherapy reduces tissue inflammation, interrupts the cascade underlying the secondary degeneration, and promotes myelin repair. Thanks to the collaboration with the Transmed research company, the GLP-grade safety of these therapeutic products has been verified in accordance with the regulatory bodies. With the IGEA company, specialized in clinical biophysics, was also tested the application of electromagnetic fields on the same in vitro and in vivo models, demonstrating extremely promising biological effects for clinical translation. Always with the objective of developing materials for neurostimulation or neurorepair, the Institute of Science and Technology of Ceramic Materials of the National Research Council has developed and characterized an injectable and electroconductive material. This material aims to promote the conduction of electric impulses and can be implanted with minimally invasive techniques in the nervous tissue. Several biocompatible components were selected. Gelatin, a biomimetic polymer able to mimic the extracellular matrix. P.PSS, able to promote the conduction of electric signals. And Genopin, a natural cross-linking agent able to promote the formation of hydrogels. The resulting samples were analyzed by rheology to optimize the gelation time and to investigate the mechanical properties of hydrogels. These results, implemented with mathematical models, demonstrated that the shear and elastic modulus of resulting hydrogels are comparable to those of the native nervous tissue. The hydrogels were then studied for the electrical conductivity, for their stability in a physiological environment, and were finally sterilized by gamma rays thanks to the collaboration of the project partner Finceramica. Mr. Smart Innovation has led the activities to study the impact of electroconductive biomaterials on brain glial cells called astrocytes. Biomaterials analyzed included electroconductive hydrogel produced by ISTEC and coated silicon nanowires devices for stimulation and real-time monitoring of electrophysiological function. The 3D hydrogel showed a significant cell adhesion and growth in the 3D and cross-linked formulations of the hydrogels, while the device recorded efficiently the functional activity of astrocytes. The second therapeutic goal of Mat2Rep was the development of new products for the repair of hard-to-heal wounds, such as pressure sores of bedridden patients, bed sores, and the diabetic foot. The CIRI SDV at the University of Bologna, starting from the results already obtained on the design and synthesis of new molecules with targeted biological activity, developed electrospun scaffolds to ensure the controlled release of the loaded molecule, GM18, which shows a beta-lactam structure able to activate some integrin receptors. The realization of this new molecule required advanced technologies, as liquid chromatography by HPLC and HPLC-MAS, gas chromatography and GC-MAS, infrared ATR and polarimetry, and nuclear magnetic resonance. To ensure a controlled release system optimized for the dressing of difficult-to-heal skin wounds, the new molecule was mounted on a fibrosis scaffold produced by electrospinning. This technology allows to control the dimensions, the internal structure, the porosity and the alignment of the fibers by acting on the solution polymer composition or modifying specific production parameters. This product was then tested at the IRET Foundation laboratories, in vitro on skin cells, fibroblasts, endothelial cells, keratinocytes, and on the pressure ulcer model in diabetic animals. These studies demonstrated that the new product is not only effective in accelerating the repair timing of the experimental wound, but also in shifting the molecular structure of the tissue towards a more physiological repair, silencing genes associated with the fibrotic type repair. The topic of skin wounds and lesions of the skin appendages, such as the hoof, is also of great interest for veterinary medicine. In the Mat2Rep working group, veterinarians studied in particular the scaffold loaded with GM18 on equine mesenchymal cells, with the aim of evaluating its translational potential in the veterinary clinical setting.
The experimental work on Mat2Rep product is still in progress. Each partner is working hard to translate these very promising results into therapeutical application in both human and veterinary medicine. Thus, I recommend to contact Mat2Rep partner to have more information and to start new collaborations.